And here's the absolutely revolutionary part.
What's that?
Biological age is modifiable.
You can change it.
Yes. And if we can measure it accurately, we can intervene to slow it down, stop it, or maybe even reverse it. For centuries, chronological age is all we had. Insurance, doctors, governments, they used it as a rough proxy for health risk.
A blunt instrument.
Very blunt. And it's rapidly becoming obsolete because we now have much smarter ways to measure biological age. The current gold standard is the epigenetic clock.
Developed by Dr. Steve Horvath.
That's the one, back in 2013. See, your DNA sequence stays mostly the same, but how your genes are expressed changes dramatically over life.
Like turning genes on or off.
Exactly. Controlled by epigenetic markers, especially DNA methylation tiny chemical tags. Horvath's clock looks at patterns of these tags at hundreds of specific DNA sites.
And these patterns correlate with age.
Astonishingly well. They can predict biological age, often within just a few years. And crucially,
Yes.
this epigenetic age responds to lifestyle. It can be reversed, at least partially, and it's actually a better predictor of mortality risk than your chronological age.
Wow. Can anyone get this measured?
Yes, there are direct-to-consumer companies now offering mail-in saliva or blood tests that estimate your epigenetic age.
That's incredible. Biological age isn't set in stone. What other ways are scientists measuring it besides epigenetics?
Well, there's telomere length, those protective caps on chromosome ends we talked about. They tend to shorten with age and cell division.
Shorter telomeres generally mean older cells.
Generally, yes. Then you have a whole range of blood biomarkers, things like markers for inflammation, like C-reactive protein, or glycation markers showing sugar damage.
And AI helps make sense of all that data.
Exactly. Machine learning can integrate dozens of these markers to give a more dynamic picture of your biological state. And beyond the molecular stuff, we have functional and cognitive assessments. These are more real-world measures.
Like grip strength, how fast you walk.
Yes, or memory test, reaction time. These physical and cognitive functions correlate very strongly with underlying cellular health and overall longevity outcomes.
Basically, if you feel old and slow,
Your cells probably feel old and slow too. It's all connected.
that makes intuitive sense. So with these tools, are we actually seeing studies where biological age is reversed? Can we really turn back the clock?
The evidence is pointing strongly towards yes, although still early days. There was a really notable, though small, study called TRIUM back in 2019.
What do they do?
Led by Dr. Greg Fay, they gave a cocktail of existing drugs, growth hormone, metformin, DHEA, to a small group of middle-aged men.
And the results?
After just 1 year, their biological age, measured by epigenetic clocks, had decreased by an average of 2.5 years.
Wow. They got biologically younger.
According to the clocks, yes. And they even showed signs of immune system rejuvenation. It was a small study, a proof of concept, but incredibly promising. It showed reversal is possible. So for you, the listener, what does this mean today? Well, there are actionable steps. First, maybe consider getting a baseline biological age test.
You know your starting point.
Exactly. Second, track your health habits diligently. Sleep, diet, exercise, stress. They all impact that biological age.
Cut out the bad stuff.
Definitely. Reduce inflammatory triggers, smoking, processed foods, chronic stress, poor sleep. Fourth, explore supplements carefully. Things like NAD plus precursors. Maybe talk to a doctor about metformin if appropriate.
And stay curious.
Absolutely. Keep learning. Keep experimenting faithfully. Longevity is a marathon. But this ability to measure biological age, it's a turning point. We can finally see age not as just a countdown, but as a dynamic score we can actively influence.
That's a really empowering perspective, moving from being passive victims of aging to active participants. And it leads us straight into the core scientific framework, geroscience. You know, we've always fought diseases one by one, heart disease here, cancer there, like spot fires.
Right, a reactive approach.
But geroscience asks a different question, doesn't it? What if these aren't separate fires, but sparks from one big underlying bonfire?
That's the core idea exactly. What if many, if not most, major age-related diseases are actually symptoms, different manifestations, of a single underlying process aging itself?
So instead of treating the symptom,
treat the root cause. Target the fundamental biological mechanisms of aging. That's the geroscience revolution. It shifts medicine from being reactive, treating diseases after they arise, to being proactive, preventive, even regenerative, aiming to delay or prevent the onset by tackling aging head-on. Now, the term geroscience is fairly new, but the thinking has roots going back decades. For a long time, the dominant view was just aging as inevitable decline, passive wear and tear.
just things breaking down.
Right. But then, starting in the 1990s, molecular biology began showing something else entirely, that aging seems to be, at least partly, programmable.
Programmable, how?
A huge breakthrough came in 1993. Cynthia Kenyon at UCSF worked with tiny roundworms, C. elegans. She found that mutating just a single gene could double their lifespan.
Wow, just one gene.
Just one. It was a revelation. It suggested aging wasn't just random decay, but maybe a regulated process that could be tweaked. Since then, countless studies have identified specific biological pathways that influence aging.
And these pathways can be targeted.
That's the key insight. They could be targeted by interventions, drugs, lifestyle changes, genetic tweaks. The field gained major credibility when the U.S. National Institutes of Health launched the Geroscience Initiative in 2013, pouring funding and focus into this area.
Okay, that makes sense. But why is targeting aging itself potentially better than our usual approach of treating individual diseases like heart disease or Alzheimer's?
It's about efficiency and tackling the source.
Sure.
Think about traditional medicine. You get heart disease, you take statins, then maybe type 2 diabetes, you get metformin, then maybe cognitive decline, another pill.
Treating things one by one after they appear.
Exactly. After they arise, largely because the underlying process of aging made you vulnerable, geroscience flips that. It goes upstream.
To the source of the vulnerability.
Precisely. If you can slow down or partly reverse the fundamental aging mechanisms, you're not just delaying one disease, you could be simultaneously delaying or preventing dozens of them.
Like fixing the leaky roof instead of just mopping the floor constantly.
That's a perfect analogy. Reinforce the hull. Before you leave the dock, don't just patch leaks as they appear. You address the core vulnerability, not just the downstream consequences. And to organize this approach, scientists developed a framework called the hallmarks of aging. Originally 9, now updated to 12.
12 core processes that drive aging.
Essentially, yes.
Yeah.
12 interconnected biological processes that seem to underlie the aging of basically every tissue and organ in the body. Let's maybe just highlight 4 really key ones that are seeing huge research focus right now.
Okay, give us 4.
All right. First, genomic instability. That's the accumulation of DNA damage. Second, epigenetic alterations, those changes in gene expression, the software noise. Third, mitochondrial dysfunction, the cell's power plants failing. And fourth, cellular senescence, those zombie cells that won't die and spew out inflammatory junk.
B cells, right.
And there are others, like telomere attrition, those chromosome caps shortening, loss of proteostasis, misfolded proteins, chronic inflammation. But those first 4 give you a good idea. Targeting these hallmarks means treating aging at its roots. And think about the societal impact. In the U.S., something like a quarter of all Medicare spending happens in a person's last 5 years of life.
Wow, that's a huge amount on late-stage illness.
Exactly. If geroscience helps people stay healthier for longer, delaying or preventing those diseases, it's not just a health revolution, it's potentially a massive economic and social one.
So, the call to action for listeners.
Adopt the geroscience mindset. Instead of just asking, how do I treat this illness? Start asking, how do I slow down the underlying biological processes that contribute to illness in the first place? That shift in thinking is maybe the most powerful tool right now.
That's a fundamental shift, all right. Okay, let's get specific now. Dive into the actual mechanisms and interventions being developed to tackle these hallmarks. Starting at the cellular level with those chromosome caps, what's the latest on telomere maintenance and TERT therapies?
Right, telomeres. Those protective tips on our chromosomes, like the plastic ends of shoelaces. As cells divide, these telomeres naturally shorten. That's telomere attrition, a key hallmark of aging.
And if they get too short?
The cell often stops dividing or dies, contributing to tissue aging. Now, the body actually has an enzyme to counteract this.
Called telomerase, or TERT.
Exactly. Telomerase, encoded by the TERT gene. Think of it as the cellular rewind button. Its job is to add DNA back onto the telomeres, maintaining their length.
So why don't our cells just keep using it forever?
Well, in most of our adult cells, telomerase is switched off or present at very low levels. The thinking is this acts as a natural brake against uncontrolled cell division, basically, a cancer safeguard.
Ah, the cancer connection. That seems like a major hurdle for therapies.
It is. It's the central challenge. How do you reactivate telomerase to lengthen telomeres without inadvertently promoting cancer? So scientists are exploring different TERT therapies very cautiously. One approach is gene therapy, delivering the TERT gene directly into cells, maybe using viral vectors.
Direct, but risky.
Potentially, yes. Immune reactions, off-target effects, and that ever-present cancer risk. A newer idea is mRNA-based delivery.
like the COVID vaccines.
Similar principle. Deliver temporary instructions, mRNA telling cells to make telomerase for a short time, might be safer as it's transient, not permanent genetic modification. Still preclinical, though.
So balancing the benefit against the cancer risk is key.
Absolutely. It requires incredible precision, maybe targeting specific tissues or using inducible systems. But for you, the listener, while these therapies are still in development, your lifestyle choices do impact your telomeres right now.
How so?
Research strongly suggests that managing chronic stress, getting regular exercise, eating a healthy diet, think anti-inflammatory foods, polyphenols, omega-3s, and prioritizing quality sleep can all help slow down the natural rate of telomere shortening.
So you can protect what you have.
Exactly. You might not reverse it dramatically, but you can definitely help preserve telomere length through healthy habits.
Okay, that makes sense. Protect the hardware. Now, moving beyond just the ends of the chromosomes to the software controlling them. Epigenetic reprogramming and Yamanaka factors. Remind us what epigenetics is again.
Sure. So if DNA is the computer hardware, the fixed code epigenetics is the software. It tells the hardware which programs, genes to run, when, and how strongly.
Turning genes on and off.
Right, using chemical tags like DNA methylation. As we age, this software gets buggy, noisy. Tags get put in the wrong places or removed when they shouldn't be. Cells start to forget their proper function.
They lose their useful programming.
Exactly. The groundbreaking idea here is, what if we could wipe the slate clean, erase that noise, and reset the epigenetic program back to a useful state? And the massive breakthrough came in 2006 from Shinya Yamanaka in Japan. He discovered just 4 specific proteins transcription factors that could do something incredible.
What did they do?
They can take a fully differentiated adult cell, like a skin cell, and reprogram it all the way back to an embryonic-like state, an induced pluripotent stem cell, or iPSC.
Wow, making adult cells young and versatile again.
Precisely. These 4 factors, OCT4, SOX2, KLF4, and c-MYC, often called OSKM or Yamanaka factors, didn't just change the cell's potential. They literally reset its biological age, erasing many of those age-related epigenetic marks.
That sounds revolutionary. Nobel Prize worthy.
Absolutely. Yamanaka won the Nobel in 2012 for this discovery.
Okay, reprogramming cells in a dish is one thing. How did this translate to potentially reversing aging in a whole living organism?
That was the next big step. In 2016, Juan Carlos's Pisua Belmonte's team at the Salk Institute tried something clever. Partial reprogramming in live mice.
Partial, meaning not turning them fully into stem cells.
Exactly. They carefully controlled the expression of the Yamanaka factors, turning them on just briefly and cyclically. And the results were stunning.
What happened?
They managed to reverse many signs of aging in the mice, improve tissue function, healthier organs, even extended lifespan in some models crucially, without causing widespread tumors or making cells lose their identity completely.
So, a rejuvenation effect without the major risks.
That's the hope, but the risks are still very real. Tumor formation, especially linked to the c-MYC factor, and cells losing their specialized function remain significant hurdles. Researchers are working hard on safer approaches, like using modified factor cocktails, maybe without c-MYC, or drug-inducible systems for better control. But here's a cool little fact from the lab. When you take old cells, like fibroblasts, and expose them to these Yamanaka factors, their epigenetic patterns shift back towards patterns seen in much younger cells.
It's like the cells remember how to be young.
In a way, yes. It suggests the blueprint for youth isn't entirely lost, just obscured by epigenetic noise. Reprogramming might help them find it again.
That's fascinating, the idea of cellular memory. Now, you mentioned reprogramming might help with another hallmark, those zombie cells. Let's talk about senolytics, clearing zombie cells. What are these senescent cells again?
Right, senescent cells. So aging isn't just about things breaking down. It's also driven by cells that basically stop working properly but refuse to die. That's senescence.
They stop dividing, but hang around.
Exactly. Usually triggered by damage or stress. But instead of undergoing programmed cell death, apoptosis, they enter this senescent state. And the real problem is they don't just sit there quietly.
They cause trouble.
Big trouble. They start spewing out a cocktail of inflammatory molecules, enzymes, and growth factors called the SASP, the senescence-associated secretory phenotype.
SASP, and that damages nearby healthy tissue.
Precisely. It creates this toxic microenvironment, contributing to chronic inflammation, inflammaging, and driving a whole host of age-related diseases, atherosclerosis, diabetes, Alzheimer's, arthritis, fibrosis, even cancer progression.
So getting rid of them seems like a good idea.
A very good idea. If we could selectively eliminate these harmful zombie cells, we might alleviate many age-related conditions at once. And that's where senolytics come in.